Hello friends, welcome to my channel AJ Lectures. So today we are going to discuss about nucleic acid. These are macromolecules for the continuity of life. Macromolecule in the sense they are made up of large number of nucleotides, polymers. DNA bears the heredity information that's passed on from parents to their children, providing instructions for how to make the many proteins needed to build and maintain functioning cells, tissues, and organisms. Basically, DNA carries genes which are the fundamental unit of heredity, and through these genes, information from parents to offspring is transferred, and the phenomena continues generation after generation. Therefore, it is considered one of the important constituents in continuity of life. So, role of DNA and RNA in cells. Nucleic acids, as we have discussed, macromolecules made up of units of nucleotides. Many nucleotide units unite and they form a nucleotides, a nucleic acid. These nucleic acids are mainly categorized in two forms. The first one is known as DNA, which is deoxyribonucleic acid, and the other one is ribonucleic acid, which is also known as RNA. DNA is a genetic material found in living organisms of all from single cell bacteria to multicellular mammals, including human beings and other animals, etc. Some viruses use RNA as a genetic material rather than DNA as their genetic material, but aren't technically considered to be alive. The reason is that viruses behave like living things in the presence of a host cell. Outside of that cell, they behave like crystals and non-living things. DNA inside the cell. In eukaryotes, such as plants and animals, DNA is found in the nucleus. So, nucleus is the region where DNA is present, and all the information to perform various cellular function is sent from nucleus because of the presence of this DNA. A specialized membrane-bound wallet nucleus is as well as in certain other types of organelles such as mitochondria and chloroplast of plants. So mainly DNA is located in the nucleus, while some amount of DNA is also present inside mitochondria as well as inside chloroplast of the cell. So the portion or DNA present inside nucleus is known as nuclear DNA, while the DNA present inside mitochondria or chloroplast is known as cytoplasmic DNA or cytoplasmic DNA. In prokaryotes, such as bacteria and DNA, is not enclosed in membrane envelope, although it's located in a specialized cell region called nucleoid. Prokaryotes, as we know that they don't have specialized nucleus, but the presence of DNA is there because these are living organisms, and the nucleus is present on a special region which is known as nucleoid instead of nucleus. In eukaryotes, DNA is typically broken up into number of very long linear pieces called chromosomes, while in prokaryotes, such as bacteria, chromosomes are much smaller and often circular in shape, ring shaped. So chromosomes inside eukaryotes, as we have discussed, is in the form of linear molecules. Linear molecules are there. while in case of bacteria and you can say other prokaryotes, this is present in the form of ring-shaped circular chromosomes. A chromosome may contain tens to thousands of genes, each providing instruction on how to make a particular product needed by the cells. There are so many genes on chromosomes, and these genes are responsible to control various characters. These genes are transcribed into messenger RNAs, and in turn, messenger RNA is translated in the cytoplasm in ribosomes to form proteins, and these proteins are responsible to control various functions of the cell. So this is the way the information or genetic information is transferred. It is from DNA to RNA to protein. This is a phenomena basically. Many genes encode proteins, as we know that, means that they specify the sequence of amino acid used to build a particular protein. Before this information can be used for protein synthesis, however, an RNA copy transcript of the gene must first be made. This type of RNA is called messenger RNA or mRNA, as it serves as a message between DNA and ribosomes. Molecular machines. These ribosomes are basically molecular machines that read messenger RNA sequences and use them to build proteins. So, in turn, this progression from DNA to RNA to protein is known as central dogma of molecular biology. Actually, the process is DNA, as we have discussed earlier, particularly in eukaryotes, is present inside nucleus. So, in nucleus, it is present. DNA is present in nucleus. The process of protein synthesis starts, and DNA, a special region of DNA, is, which is a gene for a particular trait, is transcribed into a messenger RNA transcript. This messenger RNA carries the or complete information to form a protein. Then, after the formation of this messenger RNA inside the nucleus, this messenger RNA leaves the nucleus and travels to the cytoplasm. Cytoplasm, where ribosomes are present. After reaching in the cytoplasm, they are attached to the ribosomes, and the process of translation of this message occurs, which is basically the formation of proteins. Proteins are formed. So, whole this process in which DNA first is transcribed into messenger RNA, and then this messenger RNA leaves the nucleus and reaches into cytoplasm and then attached to these ribosomes and is translated into protein, is known as central dogma of molecular biology. Importantly, not all genes encode protein products. For instance, some genes specify ribosomal RNA, which serve as structural component of ribosomes, or transfer RNAs, which are clover-shaped RNA molecules, clover-leaf-shaped RNA molecules that bring amino acids to the ribosomes for protein synthesis. Basically, these RNA also formed by the DNA, but their function is not to carry the message. They don't carry the message, but their function is different from messenger RNA. Like ribosomal RNA, we are talking about ribosomal RNA. These are the structural component of ribosomes, and these ribosomes are the sites of protein synthesis where proteins are formed. So their function is they are structural components of the ribosomes. On the other hand, we have transfer RNAs. These RNA, like ribosomal RNA, also do not contain information as messenger RNA does have, but their function is to transfer amino acids from various parts in the cytoplasm to ribosomes for the formation of proteins. So both of these have their function in the protein senses as they are the structural component of ribosomes and they are transferring amino acids, but they don't have message for the protein which are being formed. Still, other RNA molecules such as microRNA act as regulator of other genes, and new types of non-protein coding RNAs are being discovered all the times. Nucleotides, which are the uh, units of nucleic acid, DNA or RNA. DNA and RNA are polymers, are made up of monomers known as nucleotides. Basically, DNA and RNA are long chains which are formed of monomers, single units, and these single units are known as nucleotides. These nucleotides unite, a large number of nucleotides unite together, and in turn they form nucleotides in the in the form of polynucleotide chain. Each nucleotide is made up of three parts. This is important. The first one is nitrogen base, nitrogen bases. The second one is sugar, 5 carbon sugar, and a phosphate group. These three things form a nucleotide. These three things, like nitrogen bases, 5 carbon sugar, and phosphate group, unite together to form a nucleotide. The sugar molecule has a central position in its structure, as I will show you the structure in the coming slides, in the nucleotide, with base attachment to one of its carbons, and the phosphate group attached to another. These are the structures. Basically, I have told you that there are three important things in a single nucleotide. The first one is nitrogen base. Nitrogen base are of three types, and are mainly of two types. The first one is known as pyrimidines, and the second one is purines. As this is clear, these are purines and pyrimidines bases. These are pyrimidine bases. In pyrimidine category, there are three nitrogen bases. The first one is cytosine, thiamine, and uracil. In category of purines, there are two nitrogen bases, adenine and guanine. One thing is clear from the structure, that purine bases are a single ring structure, while pyrimidine bases are double ring structured. This in case of DNA, there are cytosine, thiamine, adenine, and guanine. While in case of RNA, thiamine is absent, and in place of thiamine, uracil is present. This is the 5 carbon sugar, carbon 1, 2, 3, 4, and fifth one is outside the ring. This is attachment of nitrogen base to that of carbon 5 carbon sugar. You can see that nitrogen bases is attached on carbon number 1, and this is phosphate group which is attached on carbon number 5, as 1, 2, 3, 4, and 5. Fifth, on fifth carbon, phosphate group is attached. This is a structure also showing the attachment. Nitrogen sugar, 5 carbon sugar is slightly different in DNA and in RNA. In RNA, it is in DNA it is deoxyribose sugar, as on carbon number 2 hydrogen is attached, while in case of RNA it's ribose sugar. On carbon number 2 OH group is attached. Nitrogen bases. The nitrogen bases of nucleotide are organic carbon-based molecules made up of nitrogen containing ring structures, as they are double ring and single ring structures. Each nucleotide in DNA contains one of four possible nitrogen bases. Either it will be adenine, guanine, cytosine, or thymine. Adenine and guanine are purines, as we have already discussed in the structures, meaning that their structures contain two fused carbon nitrogen rings, double ring structures. Cytosine and thymine, in contrast, are pyrimidines and have a single carbon nitrogen ring. RNA nucleotide may also bear adenine, guanine, and cytosine bases, but instead of thymine, they have another pyrimidine base called uracil. In molecular biology shorthand, the nitrogenous bases are often just referred to by their one letter symbols like A for adenine, T for thymine, G for guanine, and C for cytosine, U for uracil. DNA contains A, T, and G and C, while RNA contains A, U, G, and C. That is, U is swapped in for T. Sugar, second ingredient in the nucleotide. In addition to having slightly different sets of bases, DNA and RNA sets of bases mean difference will lie in U and T. In RNA, there would be nitrogen base uracil, while in DNA there would be thymine instead of uracil. Slightly difference is also present in sugars. The 5 carbon sugar in DNA is called deoxyribose, while in RNA the sugar is known as ribose. These two are very similar in structure with just one difference. The difference lies, as I have already told you, on the second carbon ribose, as it has hydroxyl group, while the equivalent carbon on carbon of deoxyribose does have hydrogen instead. It has hydrogen instead of hydroxyl group. The third ingredient is phosphate group. Nucleotides may have a single phosphate group or a chain of up to 3 phosphate groups attached to 5 carbon at the sugar. Phosphate is always attached at 5 carbon. Polynucleotide chains. A consequence of the structure of nucleotide is that a polynucleotide chain has directionality. It has two ends that are different from each other. At 5 prime end or beginning of each of the chain, the 5 prime phosphate group of the first nucleotide in the chain sticks out. DNA sequences are usually written in 5 to 3 prime direction, meaning that the nucleotide at the 5 end comes first and the nucleotide at the 3 end comes last. So this is all about the lecture, which was an introductory lecture about nucleic acid. So this is the basic lecture. Hopefully you will understand it, and you will read it carefully, listen it carefully, and inshallah we'll see you in the next lecture. Thank you very much.